In this video, we're going to talk very briefly about meiosis, and we're going to compare it a lot to mitosis. So if you haven't seen the mitosis video or you have questions on mitosis, you probably want to get those answered before you move on into this video, because I'm going to assume that you understand the processes that are occurring in mitosis. Meiosis is a little bit different than mitosis in that our goal in mitosis was producing 2 new cells. Well, our goal in meiosis is to produce 4 gamete cells. Not stomatic cells that are like body cells, like a bone cell or a skin cell like we would in mitosis. Humans in meiosis are going to generate either sperm cells or egg cells, gametes that are going to be used for reproduction. It's going to happen in 2 different divisions. So we're going to go meiosis I and then meiosis II right back to back. And so what that's going to do for us is it's going to take what is normally a diploid cell, which is it has 2 copies of every gene. That's how all your normal cells are. And it's going to generate a haploid cell, which has half the normal amount or 1 copy of each gene. And this should make sense because all of your DNA in every single cell is literally half of your mother and half of your father. And the only reason you got that was because your mother only gave you half of the genes she had. If each parent gave the entire complement of their genes, a diploid cell, then each offspring would have twice the amount of genetic material. And that doesn't happen. You have the same amount of genetic material as both of your parents. Half of your amount comes from dad, half of your amount comes from mom, and they're going to generate those haploid cells with only 1 copy of the DNA using the process of meiosis. We start off with interphase, exactly the same as what we're going to have in mitosis. You go through G1, S, G2. But then what happens is prophase I, and we have the formation of this thing called a tetrad. And this is very important because unlike in metaphase where these would have lined up vertically with each other, that's to say the chromosomes would have been in stacked hexes. Sorry for my bad drawing. Here, they're not stacked. They're literally together in what we call a tetrad. And that becomes very important because at this very spot right here, we're going to have something pretty special happen. And that's going to be crossing over. Crossing over basically happens because of these tetrads, and what occurs is that we're going to swap genetic material. This is really, really important because in meiosis, you're going to want to generate genetic variation. Remember that I said half of your genes are mom and half of your genes are dad. That is not to say that every sperm that dad made and every egg that mom has exactly the same halves. In fact, I would hazard a guess that every single one of them has a different composition of DNA because crossing over is going to happen all over the place. We show it happening quite simply with the tips of the chromosomes. That's not necessarily the case. It can happen all over the place on these homologous chromosomes. Homologous meaning they're the same ones. So this is 2 copies of number 1 chromosome, 1 from mom, 1 from dad. Well, prophase I is where we're going to get crossing over. It's where we're going to increase genetic variability, and this tetrad formation is a very clear indicator of a meiosis cell. If you ever see a tetrad, it's got to be meiosis, which means you're going to take a diploid cell and turn it into 4 haploid cells. Continuing, we get to metaphase I and anaphase I. You'll notice here that they're lined up in the middle again, but not vertically like you saw, but they're still kind of in these tetrads. These are pulled apart, so it's actually very, 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 very late metaphase. Really, it's a very early anaphase cell because they're pulled apart. And so what's going to happen is they're going to be pulled in opposite directions. So this metaphase I shows them in the middle. Anaphase I, they're pulled slightly apart. You can see they're not far here. They're going to be pulled apart. This is very similar to your anaphase you would have saw in mitosis, with the exception of how these chromosomes are lined up. Telophase I has cytokinesis about the same. Notice we have a cleavage furrow here, so it's an animal cell, and we have these 2 cells reforming their nuclear envelope. These chromosomes aren't going to break apart. They're not going to uncondense. It's kind of like, if you had a 2-hour stay at an airport, you're not going to unpack your bags at your 2-hour stay at the airport. You're going to leave your bags packed, pick them up, and take them to the next flight once your layover's done. Same thing here. Telophase I and cytokinesis, you're not unpacking those chromosomes. Prophase II will actually look very similar to prophase I and metaphase, and the rest of these will. It just kind of condensed because now you've kind of cut your genetic material in half. And so we're going to split these cells. Prophase I, again, you're going to not form tetrads because you don't have homologous chromosomes anymore and you pulled them apart. Metaphase II looks a lot like mitosis. So if you're looking at metaphase II, it's going to be hard to distinguish whether it's metaphase II of meiosis or metaphase 1 of mitosis, which there's only one in mitosis. And so you got to kind of know what organism you're looking at. Counting the chromosomes will tell you this. So metaphase 2, they wind up in the middle. Anaphase 2, they get pulled apart. And again, this looks a lot like mitosis. But by the end, what you end up with is 4 haploid cells, half the genetic composition. Let me flip back. You'll notice that I only show one major cell here, but I have another one in the background. That's because after your telophase and cytokinesis of meiosis I, you have 2 cells going through this. So to progress into anaphase, 2 cells are in anaphase 2, which means that instead of having 2 cells generated, you have 2 cells generated twice, which 2 times 2 is 4. You end up with 4 gamete cells. These daughter cells have half the genetic composition. So if they're humans, instead of having a total of 46 chromosomes, 23 pairs, they're only going to have 23 chromosomes, no pairs. And that should make sense because you have 46, you got 23 from mom and 23 from dad. Again, meiosis is very similar to mitosis, but the goals are different, and meiosis is going to happen twice to produce gametes.